Hi, welcome everyone. My name is Linda Isaacson, and we are here today for the EMSL Learn webinar on Pore2Chip, a Python tool for Terraforms. Before we launch into today's webinar, just a notice that our next EMSL Learn webinar is going to be on June 26th, and that one is going to focus on high-resolution characterization of mineral-associated organic matter. So we hope to see all of you at that one as well. And you can make sure that you're always in the loop on any of our events, whether they're webinars or in-person events, by staying in touch with us on our social media and email list channel. So if you have not already subscribed, please check out the link in the chat, and you'll be able to subscribe there. So for today, I'd like to introduce our presenters. The first is Erin Rooney. She is a research soil scientist for the U.S. Department of Agriculture's Natural Resources Conservation District, and she is with the National Soil Survey Center. Erin will be providing an overview of the new Pore2Chip upgrade to our Terraforms capability. Aramy Truong is a research associate at EMSL, and he will walk through how to use Pore2Chip to build 3D images of soil pores. And Maruti Mudunuru is an earth scientist at PNNL, and he will discuss the future vision of ModeEx workflows that are informed by soil datasets. There will be time at the end of today's webinar for questions. And so we encourage you to post your questions in the Zoom chat so that our speakers can answer them during the Q&A. And with that, we're going to begin with Erin. Take it away.
Great. Thank you so much, Linda. I am so excited to introduce Pore2Chip and talk a little bit about why it's important. Once I wrap that up, I will hand it over to Aramy, who will talk a little bit about the technical aspects of getting started, including installation and usage. He'll cover extracting information, creating pore networks, exporting the 2D design and fabrication options, as well as talk a little bit about how this will work for Tahoma users. And then Maruti will wrap things up for us, talking about future applications, including physics-informed machine learning, modeling, and simulations. Terraforms are a really exciting technology that allow us to visualize biogeochemistry inside soil pores. We can use terraforms to isolate and control key processes at this really fine scale resolution so that we can look at things like how soil aggregate morphology, porosity, and mineralogy interact, as well as other parameters like plant roots, rhizosphere, sorry, we're waiting for more animations to pop in, microbes and soil organic carbon, as well as nutrient interactions, and those will pop in shortly. So currently, there are a couple different ways that we can create terraforms. They can be homogeneous designs, or they can have heterogeneous and more field realistic structures, but those can be really time consuming to create. And there's something really key that those can miss, which is some pore network characteristics that are becoming more interesting to us with recent research. During my PhD, I got the opportunity to do some work at EMSL, where we looked at the effect of freeze-thaw on soil porosity parameters. And we specifically found that there was a change in a pretty critical pore throat diameter size range when it comes to water holding capacity following freeze-thaw. We also found this really interesting simplification of the pore network, where we had more singly connected pores and less very well-connected pores following freeze-thaw. And this made us really interested to know, we know that these things change, but what does that actually look like? And wouldn't it be great if we could enforce that organization onto the pores within a terraform. And then we could get a sense of water holding capacity and flow and how those things are different following freeze-thaw cycles and with these changes. Additionally, we wanted to get a better sense of how biogeochemical processes might be mediated by changes in the pore network from disturbance. Another example would be if you had some sort of ash and sediment input, how might that change pore filling, pore isolation, disaggregation, and then from there, changes potentially in microbial activity and hotspots of biogeochemistry. And so to be able to look at that, you need to be able to replicate and represent the porosity within an aggregate or soil core. So for here, for what Pore2Chip now does and adds to the ability of terraforms is to, you take a soil core or aggregate and you get the XCT data. And from there, you apply Pore2Chip, which can give a representation of these pore network characteristics. And from there, you can design and build micromodels for microbial and plant growth. And from this, you can build your own field representative reduced complexity platform. And this allows us to look at the first part of this ModeEx loop. So we can use Pore2Chip to represent pore network characteristics, and from there be able to do some pore scale modeling or upscaling using that pore information. And so now I'm going to hand it over to Aramy to talk a little bit about the technical aspects about Pore2Chip. Thanks.
Sorry, I had trouble unmuting my microphone. So thank you, Erin. I'm Aramy Truong. I'm going to talk generally more about the technical aspects of Pore2Chip, either how to make a micromodel either on your own system or to approach us at EMSL to have your soil core analyzed and a micromodel made here in our laboratory. So what exactly is Pore2Chip? Well, Pore2Chip itself is a Python-based software package that characterizes the pore network of 3D image files and generates a 2D representative pore network. Though I say software, but it's more of like a workflow. So see this top part of our ModeEx loop, we're essentially characterizing a real soil core, getting the aspects about the porosity that we want to see in our micromodel, using that information to generate our micromodel, and then finally fabricating it in the lab. And I'm going to talk really quickly just about getting started with notebooks and Pore2Chip. Since it's all developed completely in Python, the Pore2Chip is very compatible with a lot of visual scripting languages and a lot of notebooks like Jupyter Notebook. And if you have experience with Google Colab, it works great for that. And I also want to mention that XCT data or XCT image data can be very big. And while Pore2Chip does work locally on the machine, we do highly recommend that you have some supercomputing time either at Tahoma, which is our supercomputing system here at EMSL, or whichever cloud service that you want to use for yourself. We have documentation on how to get access to Tahoma, how to use it. That will be our first link up here. And we have a system called Open on Demand, which actually allows you to start a Jupyter Hub server on Tahoma with little to no setup. And I'll talk more about installing Pore2Chip on a Jupyter Hub server in a second. So here's the link for the dashboard if you have access to Tahoma. Now, I'm going to talk about building and installing Pore2Chip. At the moment, the GitHub repo is not released to the public, but it will be open source and it should be released soon. So if you would like access to the code right away, please feel free to contact us at EMSL. I'm a primary contact, and you can also contact Maruti about this. But essentially, I'm just going to go over quickly how you can install it yourself. Once we get it on the Python package repository, also known as pip, you can run it on your command line by saying pip install Pore2Chip in the command, and that will give you the latest stable build that we'll have in the repository. And if you want to build from source, that will give you all of the developmental features and a lot of the bug fixes that we'll be working on throughout Pore2Chip's lifecycle, which will be these two lines right here that you put into your command line. And we also have options for building a Docker instance. So these lines, if you have Docker installed, will help you build an instance of a program that has Pore2Chip and all of its dependencies installed, and it'll actually start a Jupyter server for you with everything installed and ready to go. And really quick for developers who want to work with our code, this will be the GitHub file structure. So all of our source code is in the SRC folder that contains all of the Python scripts necessary to edit the modules, edit to their liking. And if you'd like to contribute, that would be a great place to start. If you just want to learn how to use Pore2Chip and use it effectively, including all the parameters for the functions, there are example Jupyter notebooks in the examples folder here. And one of those Jupyter notebooks will detail how to get straight from raw XCT data to a micromodel design. And we're going to talk about more about that today. Example outputs is also a good reference because these are the testing outputs that we did for micromodels. So anything we created, we can put in the example outputs so you can see for yourself how the code should behave. The distribution folder or dist has all of our pip distribution packages. And the test folder is really good too, because that has all of the Python scripts needed to test the functionality of each of the modules in Pore2Chip. And if you're a developer and you really want to contribute, we highly recommend looking at the test folder, and it would be greatly appreciated if you see any bugs or if you would like to add more functionality to Pore2Chip. Okay, so with that all done, I'm going to talk about the general workflow of Pore2Chip and how we're going to get from XCT to micromodel. And just really quick, I want to mention that any image you see of a micromodel or an XCT image that is segmented, the black pixels are going to be representative of the pore space, which means the white pixels are going to be solid. Okay. So let's say that you start just with an XCT scan of your soil core, and we have that on the top left. And Pore2Chip has tools that will allow you to filter, do all the image pre-processing you need to get the necessary porosity data and create a micromodel. So we have a filter function that will segment your XCT image by solid space and void space simply by using certain filtering metrics that you can adjust yourself. And then once we have the segmented filtered 3D stack of images, we can use that to extract a pore networks. And this pore network is representative of the actual pore network that was in your 3D image stack. It's now a digitized node and stick system. And from that, we get all the distributions we want, things like pore size distribution, pore throat size distribution. I'll talk more about that later. But once we extract the most important bits from this network, then we use that data to inform Pore2Chip algorithm, hey, now I want to make a micromodel with these properties, right? And this workflow is very flexible. This is assuming that you only had XCT data, but let's say if you already had XCT images that were segmented in another program, or you had all the data or the necessary data extracted already, like let's say you extracted this network using a popular software called Aviso, then you can skip straight to using Pore2Chip to create the micromodel. This is just to show that you could start from the beginning if you didn't have that data, but it's very flexible. And we really are happy with Pore2Chip because it's open source. So once we have the code out, you can edit it and look at the code yourself, and you don't have to pay for a license like you have to do with Aviso. Okay, so some quick code snippets on how the data extraction works. And this is all done in Jupyter Notebook. It's very simple. Even people and scientists who have little coding knowledge can learn how to do this. So our top code snippet is going to be about inputting the raw XCT data. So it's just a stack of 3D images. And then we'll use our filtering module in Pore2Chip to extract or to segment the data into solid and void space. And there's a lot of parameters you can adjust. You can crop the image to your liking. If your XCT image is too big, which I know a lot of XCT data can be very big, you can do that or invert the colors. There's a lot more in the documentation about that. And so once we're done filtering, we have our 3D segmented image stack. And now we want to extract the properties that we're going to be using to make the 2D micromodel. So we have this, and now we're going to input it into our metrics module in Pore2Chip. And metrics, for the most part, the things that you need to create a micromodel are pore diameters, throat diameters, and pore coordination numbers. And so the pore diameters and throat diameters are rather self-explanatory. They're representative of the pore body, the main body itself, which is characterized by a sphere, and the pore throats are going to be characterizing the connections between different pores. So let's say it'll be more like a cylinder that connects those two spheres as a pore. Now, the coordination number is very important. This is arguably the most important aspect that you need to extract from the data to have a good representative micromodel. So what the coordination number is that it describes the connectivity of each pore. So let's say that you have a single pore and you have 3 throats extending from it that connect to 3 other pores. That means that that single pore has a coordination number of 3, and that number can vary depending on the different pore inside the pore network you extracted. But overall, we need to extract that using the metrics filter and the metrics module if we want to make the micromodel. Okay. And how does the data extraction work? Well, you see here on the left is a visualization of our 3D segmented images. And we're extracting the network using another wraparound function for another open source software called PoreSpy, which does all the network extraction that you would need to get the data of interest, which is the pore sizes, throat sizes, and coordination numbers. Again, if you had this data and if you had a network extracted via other means, such as a VISO, then you could skip this step, but I just want to emphasize that it's really helpful to have this open source software to be able to do all of this from scratch. And now I want to talk a little bit about what we're focusing when we make the micromodel and the things that we're not focusing on. So I mentioned we wanted the pore and pore throat information and the coordination number, which is the connectivity. But what we're not particularly focusing on are the particle shapes. And what I mean by that is we're not focusing on shaping specific grains of your XCT image or your soil, for example. So this information that we're extracting, we're going to be shaping pores and pore shapes, how the pores look and how they connect. And you'll see what I mean when we look at more micromodels in the future. But this illustrates a difference between previous efforts of making a micromodel, which would be you would pack a bunch of spheres together and then take a slice of that sphere packing, and you get a somewhat heterogeneous looking pore network, but it didn't capture the pore connectivity and the pore aspects that you would get with using our method. Another thing I want to mention are ferrite diameters. If you don't know what ferrite diameters are, it's just a measure of an object along a certain axis. So if you had an oblong shape, a ferrite diameter measurement on one end would be one length. And then if you measure it from a different angle, let's say a perpendicular axis, it may come up with another angle or another measurement. And this is useful for characterizing the specific shapes of pores and pore throats. And I want to mention that we're not particularly focusing on ferrite diameters when making the micromodel. However, Pore2Chip does have the functionality to measure ferrite diameters and plot their distributions like I have down here of your segmented XCT image data. So it is very useful for user reference to see, okay, what are the general minimum and maximum shapes of my XCT data are. And it's just good to know once you start creating the micromodel so you can compare. Another thing that we're going to focus on a little bit later is the overall porosity. Now, you can change the overall porosity of the final micromodel, but it's not the biggest focus that we have when we're creating the micromodel right now. Okay, so now let's say we have all the necessary data extracted. We have the pore diameters, throat diameters, and coordination numbers. Now we can start building a representative 2D network. And we're going to do that using another open source software. And all of this is going to be built into Pore2Chip. And this one is called OpenPNM. And what OpenPNM does is that it allows you to create digital networks that will simulate pore networks. like the one you see below. So what you see right here, and again, red dots are going to be the pore bodies and the blue lines are going to be the pore throats. And what we do is we take the data that you extracted, so the pore diameters, we get their distributions. So what is the likelihood a pore would be this diameter, for example? And then we feed that to the pore-to-chip algorithm, and then it'll create an open PNM network that has the pore diameter and throat diameter distributions that we extracted from before. And the same with coordination numbers. So what we have here is a diamond cubic lattice shape that has been tuned with the parameters that we gave it. And it's not visually shown here, but each red dot has its own diameter assigned to it. Same with the blue throats. Those will have their own diameter assigned to it. And also we have all the coordination data that we extracted. So before, let's say if we extracted coordination numbers from the XCT and there were a lot of pores that had a coordination number of 4. And so we see that in the micromodel, there are pores with 4 connections coming out. And that shows that we are capturing the connectivity of the XCT images in the final micromodel. And another thing to mention, the pore positions, these red dots, are slightly randomized to not have it be a perfect cubic shape. You can change that factor to randomize it more or less, or have it as a straight cubic shape if you want. That's just an option that we have with pore2chip. And the really good part about this is that everything is scalable when it comes to generation. So these different open PNM networks were generated using the same data. We just specified the number of pores that we want in general in our micromodel. So let's say, for example, I could create a micromodel that just had 5 by 5 pores, or 10 by 10, or 15 by 15. You're not limited by the amount of input data you have to make a different size output data, so to speak. Okay. And so once we have this scaffolding, now we're going to actually draw the micromodel. And this is what we're going to be using when we fabricate or do your simulations on. So on the left, we have an example of an open PNM network with all of its values assigned to each pore. And then what pore2chip does is that it uses a drawing Python module to draw images of the open PNM network with the correct pore diameters attached to them. So let's say this pore right here had a diameter of 2 units, let's say. And now we're going to draw a sphere or a blob in that position that has a diameter of 2 units. And we do that both for the pores and the pore shapes and the throats. You could adjust the pore shapes if you want. In this image, they are randomized blobs, but you can also change them to be circles. And one thing I want you to notice is that a lot of the throats, they're not straight lines. So they're not like cylinders, like when we extracted them. There are features of pore2chip that allows you to change the tortuosity of the pore throat, or the general like waviness of the pore throat. And you can adjust this too. It'll be a random amount. If you wanted straight throats, you can change the tortuosity factor to 0 if you'd like. And on the bottom right, here's an example. This is just a mock-up of this example micromodel that I created in SolidWorks. So this image that we have here, this vector image, can immediately be used to create STL files or any 3D file format you want if you want to make this with additive fabrication, such as 3D printing. And it's just a proof of concept to show that everything works. And since it's a vector format, you don't lose detail when you zoom into the image. So in this case, it's a SVG image that we export. I brought it into SolidWorks and made an STL from that. And here I'm going to mention that overall porosity thing again. So when we're creating the micromodel, we don't really know the final porosity of the micromodel, but we can control it by adjusting the number of pores in the micromodel or changing the size of the image of the micromodel. So for example, if I wanted less porosity and I want to change this network, instead of doing 5 by 5, I can do 4 by 4, and it'll move those 4 and stretch it out within the same distance. So you have the same pores of the same diameters and the same throat diameters, but they're just more spread out, and that would decrease your porosity. And same goes vice versa. So if you had analyzed your XCT data and it said it had a porosity of 10%, let's say, and your micromodel had a porosity of 20% and you wanted to adjust that down, then you could do what I just mentioned, which is changing the pores or the image size. It is a lot of technical work, and I'm going to take this moment to highly recommend that if you are using Pore2Chip yourself, come talk to us at EMSL, or even just have us at EMSL do the adjustment for you, and we can go back and forth about how to tune your micromodel to your liking. Okay, and again, just like the network that we generated before with OpenPNM, all of these micromodels are scalable. So you're not limited by your input data. And these are in units right now because we extracted the data from the XCT image in terms of pixels, but then you can scale these to whatever dimension you want. So if these were 50 by 50 micron, for example, you can print that or have it etched with no problem since it's a vector image format. Same with the 500 by 500. It's very flexible. Now, earlier, my colleague Erin mentioned the effects of pore disturbance, and particularly she mentioned her research on freeze-thawed soil cores. So on the top left and on the top right are example images, but these are from actual soil aggregates. And Erin, you can correct me if I'm wrong, but I believe these were from a field site in Alaska, where one soil core was normal, and these soil pores have some water in them. And then they did a test where they froze the soil aggregate and then thawed it out just to see what the effect on the connectivity was. And that's what the XCT images on the top right are. And now what we did here at EMSL is that we generated some micromodels to see if we can capture the connectivity change between the normal soil core and the freeze-thawed soil core. And you can see on the bottom left, this micromodel, which represents the normal soil core, had a lot of general connectivity. There were not very many disconnected pore nodes and not very many singly connected pores. But after, when we generated a micromodel that represent the freeze-thawed soil core, you can see that there are a lot more lone pores and a lot more singly connected pores. And this is exciting because it shows that Pore2Chip can really capture a lot of the specifics of your general or your specific XCT core in terms of connectivity and pore sizes. And we're really happy to see that. And that's why we are really pushing why this is such a great technology, because you can make representative pore networks of any XCT, and it'll be unique to that specific one that you extracted data from. Okay, so now I'm going to talk about fabrication and all the ways that we can fabricate physical micromodels in the lab. So here at EMSL, we have two ways to make micromodels, and one of them is laser etching and another is photolithography. So in the images you see right here, especially in the top left one, this is a 3D print of the pore space of a soil aggregate, and in front of it is an acrylic glass with a micromodel actually laser etched in. And here's the same image on the bottom right where a researcher is holding it to the camera. And these were made using laser etching. So we have a laser etcher that can make micromodels in maybe 20 minutes, even less. It's very high throughput process, very efficient and rapid for prototyping. And this time could be even saved more if you're etching more than one micromodel at a time. So laser etching is very good. It captures good detail down to about 15 micron or so here at EMSL. If you have features in your micromodel, let's say that your micromodel had a throat that is less than 15 microns wide, then we could use the laser etcher, but we would highly recommend using photolithography at that point, which gets detailed down to 1 micron, and it takes a long time, but it's the most accurate. So there are pros and cons. We're really happy about the laser etching capability that we have because it'll make things really fast. And we highly recommend talking to us about specific options for your XCT images or your soil core and what options you would like to use when making a real physical micromodel. And one thing to mention is that in these images, we etch them on acrylic glass, but most likely we'll be making micromodels on epoxy resin slides, which I'm going to talk about now in a second. So when you come to us at EMSL and you want to make a micromodel, it's going to start with a glass cover slip with some Indian tin oxide coating on it, which allows it to be conductive. And above that is going to be the medium that we're going to use to make the micromodel. So it'll be maybe epoxy resin. We use laser etching on the surface or photolithography. And this is where the micromodel design will be. And once we're done with that, we put a membrane or some sort of PDMS cover on the top, and you have your micromodel habitat ready to go. And you can do things like grow fungi, grow microbes in it, or even if you scale it, like we talked about before, you can even grow plants and plant roots. And Terraform or the Pore2Chip platform is very compatible with a lot of EMSL technologies, including characterizations like biochemical assays, microbial and plant root biomass omics, optical microscopy, and in situ chemical imaging, which includes things like MALDI or nanodesi or SIMS or electron microscopy. So I'm very happy with the system we have here, and I'm excited for what we can make with these micromodels. And now I'm going to pass it on to Maruti, who's going to talk more about the future development of Pore2Chip.
Thanks, Aramy. And today we'll be talking about building on the Pore2Chip platform. Within the Pore2Chip platform, our team is developing advanced modeling techniques to simulate numerical simulate complex multiphysics phenomena. So these numerical simulations are essential to understand and predict how the carbon flows and moves within this micromodel, and how the nutrient intakes within this micromodel for various applications related to the rhizosphere function or soil organic carbon cycling. So the micromodels that are generated within the Pore2Chip can be meshed. So what I'm showing here is a mesh that is generated by the Pore2Chip for multiphysics simulations to better understand carbon flow and reactive transport modeling. So a structured mesh is a capability that is needed to generate flow and transport simulations for p-Floetran-based multiphysics modeling. And the mesh is a set of grid points to solve carbon flow and reactive transport within the lab-on-chip application. So the key modeling capabilities that our team is developing include the physics-informed neural networks. And the physics-informed neural networks are the networks that can allow us to integrate physics laws such as Darcy's flow, diffusion equation, advection diffusion equation with experimental data to enhance machine learning modeling capabilities and make it much more predictive and reliable. In addition to that, we also provide p-Floetran input decks as a part of our future application. p-Floetran is a massively parallel subsurface flow simulator that is widely used in the DOE complex to simulate movement of fluid flow and carbon degradation within this porous micromodel. In addition to that, we also are providing CFD simulations and associated tech. So CFD simulations are utilized to simulate fluid flow within the micromodel, how the fluid flow goes within these pore spaces that Aramy has mentioned, within these black spaces. So CFD is utilized to simulate this fluid flow within the micromodel, providing additional insights on how the dynamics of the fluid flow movement and how it interacts with the solid matrix within this micromodel. So what I'm showing is a detailed mesh here that is given as an input both for the CFD simulations and p-Floetran input deck simulations, along with the physics-informed neural networks where these meshes are used as a part of the collocation points. So as a next step within this modeling examples and capabilities, so what are we showing is a future vision of this ModeEx loop. So what is this ModeEx loop? So as described previously by Aramy, the ModeEx loop, the model, experiment, and data integration loop, represents a comprehensive approach to integrate modeling and experimental data to refine our understanding of carbon cycling within the soil course through these micromodels. That's the primary reason, one of the primary reasons why we are generating these micromodels. The ModeEx loop includes various key steps. So let me quickly use a laser pointer. One second. So the first step, as Aramy pointed out, we have the XCT imaging of the soil cores. So the high resolution XCT images provide information on to capture detailedly the 3D structure of the soil cores and aggregates. So this step provides a foundation understanding of the soil structure property relationships needed for the upstream or the downstream modeling for pfloat RAM or any other physics informed based neural network modeling. So the first step provides that information. Once we get that information from the XCT imaging of the soil core and aggregates, the second part, as Aramy described, is a 3D pore network characterization, in which we get more information on the 3D pore network structure, the associated coordination numbers that are needed for the understanding of fluid transfer properties in the porous media. And once we have that, we transform that and provide a 2D rendering of that and then build the micro models. So the capabilities we are trying to develop are in the step 5, 6, and 7. So the step 4, once you build a micro model, we perform experiments to conduct detailed analysis of how the carbon flows and how the carbon is being degraded, what are the associated observations with respect to the carbon flow and degradation phenomena. So once we get this experimental data, so what we do is within this experimental data, we get a detailed understanding of how the chemicals hotspots are formed within this porous media. So techniques such as time of flight secondary ion spectroscopy, so that is a TOF SIMS, and the SEM EDX, that is a scanning electron microscopy with the energy dispersive X-ray spectroscopy, are used to understand and analyze these hotspots. So once we get a better understanding of these hotspots, that is ingested into the process models that is given in the step 6. So in the step 6, we perform detailed pore scale multiphysics modeling. And in this multiphysics modeling, we model fluid flow, specifically the carbon flow, heat transfer, and the chemical reactions within the porous media. And these multiphysics porous models help us in understanding the predictive behavior of the soil carbon cycling under different environmental conditions. So that is the objective of this step 6A. And once we get that information, we use that data to calibrate the process models either in pfloat or CFD simulations or physics informed neural networks. So these models are calibrated using this experimental data in the step 5 and step 4 to ensure that they are accurate and reliable. And this step is essential for refining the multiphysics process models for better understanding the soil cores and improving their predictive capabilities. So that is the calibration and validation of the multiphysics process models. So once we get this information tied up and calibrated through the step, we use that information for better understanding the carbon flow and degradation in the bigger soil cores. So experiments are conducted to better understand how the carbon flows and degrades within the soil core to the hydraulic experiments. So this is vital for better understanding the carbon cycling and rhizosphere function if you have roots, and connecting back to the micromodel generation. So it's an iterative loop. So what we understand from the micromodel is transferred back to the soil core, and what is observed in the soil core is transferred back to the micromodel in an iterative fashion to update this ModeEx loop. So once we get a better understanding of that, this is upscaled from the pore scale at the micro scale to the soil core, that is at a millimeter and centimeter scale, to better understand the findings for real world scenarios and for large environmental systems. So this iterative ModeEx loop ensures continuous improvement of this multiphysics process models and better calibration of them, so that we can transfer that information going from the pore scale to a macro scale to a continuum scale to a watershed scale, leading to a better, accurate, and reliable predictions of soil carbon cycling at various different scales. So this capability is now is available for EMSL users. So EMSL exploratory call is open, and we would like the users to apply for this call to utilize this capability. So primarily, this exploratory call is for 9 months, and this is an excellent opportunity to create your field represented micro models and test them, and develop experimental databases and use that information to perform physics informed machine learning for better understanding of the soil carbon degradation or understanding the rhizosphere function. So with that, we conclude and we are open for questions. So back to you, Linda.
Hello, all right. Thank you, everyone, for your excellent presentations. I hope everyone in the audience was able to learn more and get excited about what's to come with Pore2Chip. So Heather, do you want? Excellent. Thank you. So we did get some questions from the audience, and of course, at any time, if anyone has a question, feel free to drop it into the chat. We will grab it and get it answered for you. So the first one, this one comes from Andy Lipton. Will Pore2Chip work with other 3D imaging techniques, such as MR imaging? And what are the resolution limits?
Yeah, I can answer this one. There really is no specific resolution limit, and whichever 3D imaging technique you use, it should work. I would note that we would turn your whatever image you have to grayscale, because that way then we can segment whatever you have into pore space and solid space. Obviously, the higher resolution, the better, so you can capture more details of the pores and the pore throats. And I talk a lot about XCT imaging, but it should work with other imaging techniques too, like MR imaging or even OCT images. So yeah, there's no specific resolution limits. And once we get the code out, you can also experiment for yourself to see what works and what doesn't.
Great. Okay. So Pedro had well, not so much a question. I'm more like a statement, but I do think, Aramy, if you could answer. He says, I didn't understand the images shown in the plots, if they are a slice of the soil column or an artificial representation of it.
So I wish I could go to the back of the slides, but there were some images of the XCT images that we're doing testing on that were actually slices of a test soil aggregate. So it wasn't like a specific soil core from any specific field site, but those were just a representation of the 3D images that I just sliced for this presentation. And if you're talking about the images with the red dots and the blue connecting them, that is just a digital open PNM network that is a 2D representation of it. That's what we're generating when we want to make a representative micromodel, if that makes sense. I apologize if I can't answer that right now. I'd like to have the images in front of me, but yeah.
And if I could just add really quick, and I'm sort of making an assumption about what the question is targeting. But, oh, it was about the network. Okay, great. One thing I just wanted to add in terms of the history of Pore2Chip is that before Arunima and I got to work with this great team and Aramy and Maruti, our initial plan was to take a slice of the XCT, just select a random slice of the hundreds of XCT 2D images that go together to make the 3D network. And that was really what highlighted the need for Pore2Chip, because if you just select a random slice, there's no way to guarantee that you're going to get the representative characteristics of that pore network. And you could kind of see it where Aramy showed the freeze thaw, before and after freeze thaw. And in the slices that were shown, you could kind of see the difference, but it was in the representative pore networks from Pore2Chip that you could really see the differences. Thanks.
Great. Okay, we have another question from Arjun. And I think this one is for you, Maruti. How computationally expensive reactive transport is reactive transport simulation on a size of 1 centimeter squared chip would be? And what are the advantages of using pFlowTran as a simulator versus open PNM?
Okay, so let me answer the first question. So that's a very good question. So we are working on developing the input decks for the pFlowTran. So pFlowTran is highly scalable. An equivalent representation of that in a different context, we are able to deploy that on Tahoma and simulate a steady state and a transient problem with within a couple of hours. So the entire simulation took within a couple of hours for approximately 10 million mesh cells. So you can go up till 2 billion mesh cells, and that can be easily be scalable and addressable on Tahoma machine. And the second question, advantage of using pFlow versus OpenPNM. So OpenPNM is like a first order fidelity model. So think like it's a, if you want a high resolution simulations, OpenPNM cannot provide that information. pFlow can provide that information. So if you want to get a first understanding of a quick and first and a low fidelity understanding of the flow, then you can get that information. But OpenPNM, if I understand correctly, may not be able to do the entire carbon flow and degradation. So pFlow has representative models to do the carbon degradation through a reactive transport model and a sandbox. So in that way, the advantage of using pFlow over OpenPNM is that like pFlow provides much more capabilities compared to OpenPNM, and OpenPNM can be used a quick way to get a better understanding of the fluid flow. Hope that answers that question.
So the next question from Maz. Does EMSL have their own modeling framework, or do you provide the modeling as a service on open foam, for example?
Okay, let me take that question. Oh, that's a very good question. So at EMSL, we provide service. Like for example, a user comes with a request saying, "Hey, I have this pore2Chip micromodel etched. Can you help us with the pFlow or open foam simulations?" We can provide that as a service. Yes, through the user proposals. That's the reason why we wanted users to apply to the EMSL exploratory proposal call. That will help us to provide that modeling as a service to the users as an example on the pore2Chip. Hope that answers your question, Moaz.
And Tina, I think, has another one for you as well, Maruti. Has the whole ModeEx loop been validated between 3D XCT and 2D chip situation? I.e., is the error between 3D and 2D known? Maybe this is a Maruti, or maybe this is Erin or Aramy.
So maybe let me take the first cut, and then Erin and Aramy can chime in. So the ModeEx loop, the pore2Chip, we have constructed the first 4 steps in the figure. The entire ModeEx loop has not been validated yet because we are still constructing the modeling applications for the 2D micromodel, and then have that experimental data ingested into that. But at least when we release the GitHub, we'll have our team members have developed the physics informed neural network capabilities to perform flow and transport simulations on that. So that capability will be advertised in the GitHub and the subsequent EMSL proposals towards validating the ModeEx loop. So answering your question, it is not there yet, but that is our aim of using the experimental data from the 2D model simulations and to experimental data and going back again to 3D. So I'll let Erin and Aramy chime in if I'm saying something different.
No, that's completely right. And with the error between 3D and 2D, I think that's more of a simulation question. I'm sure Maruti could answer later. But there are some things that have to be generally lost when you go from 3D data to 2D data. That will really depend on a lot of factors, and also depends on what you're analyzing. So what properties are from your XCT and what can we bring to the micromodel. So it's really dependent in that aspect.
That's correct, Aramy.
All right, next question. How can I create a RhizoChip? You talked about micromodels.
Yeah, so I can answer this one. Really, a micromodel is a RhizoChip. It's just a very scalable RhizoChip. So in that slide, when we talked about manufacturing a micromodel, it's essentially the same thing as a RhizoChip where you just have pillars that represent the solid space and the area in between. That's the pore space. And Pore2Chip just gives us another way to generate that micromodel or that RhizoChip. And since it's scalable, you can make it big or small. I know RhizoChips are usually used for testing plant growth and plant root growth, and you could do that the same way with Pore2Chip. It's just a matter of scaling.
Great. Okay. How do you use this Pore2Chip capability in an EMSL user proposal? Erin, do you want to take that one?
Yeah, I can take that one. So that is kind of the extent of my experience as an EMSL user, and the development of this was to support other EMSL user proposals. I would say that something to keep in mind is what Pore2Chip can give you, and whether or not that links with the hypotheses and the research objectives of your proposal. So if you want to understand maybe a change in pore connectivity, if you have a hypothesis that and you're planning to get information about pore connectivity, and you have a hypothesis for how that might change and what that change may do in terms of potentially biogeochemistry, if you want to link with Maruti on the modeling, that would be an appropriate use of Pore2Chip. Same with a change in pore throat size distribution, pore size distribution, and then as Aramy mentioned, to some extent, changes in pore volume, although that's going to be secondary to the other capabilities of Pore2Chip. So I would say that any environment or any, and something that I think we also want to emphasize is that it's not always, or it doesn't always have to be soil. It can be any porous media that you can use Pore2Chip for, to get a sense of how changes in connectivity, changes in pore throat size distribution, and pore distribution can impact some sort of dynamics within that chip. Is that fair to say, Aramy? Maruti, do you have any additions to that?
Yeah, that sounds about right.
That sounds right, Erin.
All right. Let's see. How do these, how to use AI capabilities for different codes and data analysis? Can you provide any examples?
So let me take that, Linda. So once the Pore2Chip GitHub release will be available, it will be available to the EMSL GitHub. So you can download based on the instructions that Aramy has provided, and we have provided some example Jupyter notebooks and Google Colab notebooks, so that you can run the physics information learning or the deep neural networks that our team has developed to understand flow within this synthetic micromodels, and see how the flow happens, how the transport happens. So, and once you test those capabilities, you can provide that feedback to us and reach out to us through either to the point of contacts that is listed in the GitHub repository, or you can apply to the EMSL exploratory research to advance these capabilities in a way that will help not only for your research questions, but also overall scientific community. So we have provided preliminary examples, but as Aramy mentioned, the best way to contribute is to understand through the testing folder. So create more functionalities for the test and make sure that these capabilities are not only important for you, but also for the overall scientific community. Hope that answers your question, Linda.
Yep. All right. How does the oblong pore shape retail all of the pore characteristics from the soil core, Aramy?
Yeah, so in those example micromodels that I showed, we made pore bodies that were of a sort of oblong or blob shape. And how Pore2Chip works is it still keeps the diameter that you extracted from the XCT core data. So even though it's maybe a more blobbish shape, it still retains, if you measured it, the actual diameter that you extracted from the XCT images. So, and of course, if you don't want it to be a blob, you can always change the pore body shape to a circle, and that will definitely guarantee that, hey, this measurement from one end to one end is the same diameter that you got from whatever you analyze, soil or whatever core.
Okay. Well, talking more about that, how do you deal with right angles that are seen in the micromodel design?
Yeah, so we might have seen some in that example open PNM network with the red dots and the blue lines. And those are generally an artifact of us using a cubic diamond lattice shape for our pore network. And we can mitigate that by changing a lot of the parameters for generating that network, like the randomization of the pore positions. So I mentioned you can change the amount that the pores are randomized positionally. If you increase that amount, it'll change them a lot more and it'll look a lot more drastic. And you might have seen that when I showed the freeze thawed and the normal soil aggregates where I changed the threshold a lot for randomness. So it doesn't look like complete 90-degree angles. And another thing to mention too, if you're worried about making the physical micromodel, is that these right angles are not very apparent when you make the actual micromodel, whether it's laser etching or photolithography.
Okay. We have just enough time for just a couple more questions. Aramy, since you're talking about the design, let's do one more in that vein. How do you control the overall porosity of the micromodel?
Yeah, I mentioned a little bit before, and I'm going to preface this with it's a lot of technical work and trial and error and a lot of forethought knowledge about your specific micromodel and XCT image, which is why we recommend you visit us at EMSL about it. But essentially, like I mentioned before, you can change parameters about the size of your micromodel. So if you kept the number of pores the same and you increase the size, it'll spread out those pores more, and they'll still have the same diameters and throat diameters that you assign, but they'll be more spread out. So there's going to be more white space, and that white space is the solid space. And the same vice versa if you decreased and you kept the same amount to increase your porosity. And of course, you can always change however number of pores that you want in your final micromodel. We leave this all up to the user because it will really depend on what the user wants to do with their micromodel, whether it's fabricating for in real life lab on chip experiments or running simulations like Maruti was talking about. So it's very flexible when it comes to changing the porosity.
Great. And our last question is for you, Maruti. If I need help on simulations and modeling on micromodels or ModeEx, who should I reach out to?
Yeah, that's a good question. So it's similar to what Tina asked. So you can just reach out to us. So we have the point of contacts here. We have Erin, Aramy, myself, another couple of developers for the pore2Chip. So reach out to us. Once the GitHub release is done, you'll be able to see a lot more people contributing to this pore2Chip. So we are excited to get feedback from you and contribute to this repository. So don't feel shy. Come talk to us.
Yeah. All right. Well, that concludes today's webinar. Want to thank everyone for joining us, and don't forget to that we will have our next EMSL Learn webinar on June 26th on high resolution characteristics of soil organic matter, mineral associated soil organic matter. So join us then. And yeah, thank you for stopping by and learning about pore2Chip.